So when Mendel's work was rediscovered in 1900, the race was on to find out where in cells were genes or factors for heredity located. Mendel did not know where the factors were located. He didn't even know about meiosis or mitosis. In 1900, mitosis and meiosis had been observed, but no one looked at them in great detail. In 1902, Walter Sutton, observing spermatogenesis in grasshoppers, and by the way, spermatogenesis and oogenesis are very difficult to observe because the cells are so fragile they usually die. Um, Walter Sutton, watching the chromosomes separate during spermatogenesis, hypothesized that it was the chromosomes that contained the genes. And the chromosome separation was similar to the law of segregation that Mendel proposed in his paper. And the theory was born in 1902 that genes are on chromosomes. Now, if that's the case, it quickly became realized that a chromosome is not a single gene. A chromosome contains hundreds or thousands of genes. Genes that are on the same chromosome do not undergo independent assortment and meiosis. They're inherited together as a group. If you're playing cards, the two of clubs and the two of diamonds are two separate cards, and they can be handed out separately, but the top of the two of diamonds and the bottom of the two of diamonds are part of the same card, and they go together no matter what. They cannot separate. Well, that is the case for genes on your chromosomes. So the study was on to try and figure out how do cells get around genes being stuck together on the same chromosome. These genes being stuck together are called linked genes. Linked genes were first discovered in fruit flies by Thomas Hunt Morgan right after Walter Sutton observed meiosis in spermatogenesis of grasshoppers. So what Thomas Hunt Morgan did when he discovered linkage was he crossed gray normal-winged flies with black small-winged flies. And the small-winged are basically vestigial winged flies. The wings don't work. They're tiny. Now, he should have gotten, from this cross, if you do a Punnett square, you should get one gray small-winged fly to one gray normal-winged fly to one black normal-winged fly to one black small-winged fly. But that's not what he got. When he crossed these two flies, he got enormous amounts of gray normal flies or black small-winged flies and very few of the others. His conclusion was that the genes for the body color and the wing type were on the same chromosome. And so the gray normal winged flies were mostly handing down the gray normal chromosomes, and the black small winged flies were handing down black small winged chromosomes. The only reason he got some flies that were gray bodied with small wings or black bodied with normal wings was because of crossing over. He hypothesized that there had to be some process going on in sperm formation and egg formation that allowed the information from the separate chromosomes to swap over. We call that crossing over. You learned about that in meiosis. Morgan got the Nobel Prize in 1933 for his discoveries on inheritance using the fruit flies. So what crossing over is all about is basically it's allowing linkage groups to be broken up so that chromosomes don't just always have the same genes on them, that they get different genes. That is one of the special things about sexual reproduction. Your chromosomes that you were given by your parents are allowed to exchange pieces. So in you, your mom and dad chromosomes can trade pieces and become unique chromosome types, not because the genes are different, but because the combinations of genes are different. And Thomas Hunt Morgan discovered it is possible to map genes on chromosomes using recombination ratios, which we're not going to go into here, but he did that, and that was part of the reason he got the Nobel Prize. Now, another peculiar phenomenon of inheritance is sex linkage. In 1905, Nettie Stevens discovered sex chromosomes. Sex chromosomes, when there are two X chromosomes, are homologous, but an X and a Y are not homologous to each other, at least not fully. Humans have 44 autosomes. Autosomes are non-sex chromosomes, and 2 sex chromosomes. So because humans have 46 chromosomes per cell. 44 of the chromosomes are autosomes, meaning they don't determine gender. 2 of the chromosomes determine gender, and they're called sex chromosomes. In sex determination, females get 2 X chromosomes, that's why they're female. Males get 1 X and 1 Y, that's why they're male. It turns out males determine the sex of their offspring at conception, depending on whether they hand down the X or the Y. Now, it turns out that the sex chromosomes carry a lot of non-sexual information on themselves. And so there are many genes on the sex chromosomes that play no role in determining sex, but they do determine the health of the organisms. Such genes are said to be sex-linked. And in fruit flies, eye color is sex-linked. In humans, muscular dystrophy, color blindness, hemophilia are all X-linked, sex-linked diseases. Now, Thomas Hunt Morgan discovered this phenomenon in fruit flies. He crossed a red-eyed female that was heterozygous with a red-eyed male. And this is the cross he performed. Now, notice we're using X and Y. The XY represents the male fruit fly. The XX represents the female. The female was heterozygous. One of her X chromosome had the red gene. The other one had the white gene. The male, his 1 X chromosome has the red gene. Notice that his Y chromosome has no eye color gene at all. What Morgan discovered was that the fruit flies faithfully produced a ratio of 3 red to 1 white in the offspring. That's what you would predict by Mendel's genetics logic. The thing is, though, all of the flies that were white-eyed, that's recessive, all of them were male. And that showed to Thomas Hunt Morgan that the eye color genes for fruit flies are on the X chromosome. Once Morgan had pinned that down, it became clear then why human boys get muscular dystrophy but not girls, why boys almost always are more frequently colorblind than girls, and why boys get hemophilia but not girls. It has to do with the inheritance by males of only one gene, sex-linked gene, which, if it's bad, produces a diseased offspring. For a girl to get hemophilia, both of her X chromosomes would have to have the defective blood clotting gene, and that would truly be rare. Now, chromosomes do occasionally mutate. If the whole chromosome gets screwed up, we call that a chromosomal mutation. If just a specific gene on the chromosome gets screwed up, we call it a gene mutation. Chromosomal mutations usually are kind of catastrophic. If part of a chromosome is missing, we call that a deletion. If part of a chromosome is doubled, and that happens rarely, that's called a duplication. Sometimes sections of chromosomes are upside down. Those are called inversions. And occasionally, especially in cancer cells, part of one chromosome has been moved and is attached to another chromosome. Those are called translocations. These mutations tend to be rather harsh. Usually, cancer cells exhibit deletions, duplications, inversions, and translocations. And that's why they became cancer cells. That's pretty common. But it turns out babies also get born with serious genetic difficulties because of deletions or duplications or inversions or translocations. Now, there are also mutations that only involve a single gene on a chromosome. If it's just a single codon of the gene, if it's just a single nucleotide, we call that a point mutation. Point mutations can be serious, but they often are silent, meaning they don't cause any problems at all. Because remember, some amino acids are represented by up to 6 different codons. So sometimes changing a nucleotide in a gene doesn't even change the genetic message being expressed in the protein. Frameshift mutations are worse. If a frameshift mutation occurs, usually it wipes out the gene. What a frameshift mutation is one of the nucleotides gets lost. And so the whole gene gets shorter. And that shifts the reading frame. The codons are read every 3 nucleotides. If you pull out one base pair or nucleotide pair out of a gene, the whole gene gets shorter, and now the whole codon sequence reads differently, and a whole different set of amino acids will get transcribed. And that's sort of disastrous. So generally speaking, frameshift mutations are almost always worse than point mutations. So here's chromosomal mutations. Here's a normal chromosome, but then this section got deleted. Now it's shorter. That's a chromosomal deletion. Here's a normal chromosome here, but then that section got over-replicated. It happens. And you end up with a super long chromosome. That's a duplication. Here's another one where you got this section of this chromosome here, and for some reason it broke out, it flipped around, and then it reattached, and now it's upside down. Sometimes those are a problem. Occasionally they're not a problem, occasionally. And then sometimes this piece of this chromosome breaks out, so the whole yellow chromosome gets shorter, and then this chromosome gets longer because it picks it up. That's rare. That's the translocation. That's what we talked about right there. So chromosomal mutations are thankfully rare, but they're very common in cancer cells. But they are almost always worse than simple gene mutations. Now, genes talk to each other and they interact with each other inside of cells. As it turns out, recessiveness is really not, it's not a matter of being submissive to another gene. Recessive genes are usually defective and they don't make a protein product. That's why we call them recessive. They're just not working. The functional gene that's working is then what we call the dominant gene. In incomplete dominance, in 1760, Joseph Kohlreuter was crossing red and white carnations, and he found in the offspring a quarter red, a half pink, and a quarter white offspring. He got Mendel's ratios 100 years before Mendel, but he could not explain how they worked. And so he doesn't get any credit for discovering heredity. Mendel's genius was he actually modeled with mathematical models how it all would have worked out. We now know that the carnations, the white carnations, have no functioning red gene, and as a result, the flowers by default are white. Now, if you are a heterozygous carnation and you have a red gene and a white gene, you make some red pigment, but not enough, and that's why you're pale red or pink. If you're a carnation and you have two red genes, now you're cranking out a lot of red pigment, and that's why you're red. Codominance also is not just because two genes are both, dominant to one another and they're both beating each other up. It's just that both genes are making a protein product. And as a result, in blood type, if you're type AB, which is rare, that means your body is manufacturing the type A blood cells and the type B blood cells. And as a result, you're type AB. That is the nature of codominance. On the other hand, type O blood type is recessive. It doesn't actually have a marker on it. So the blood types are an excellent example of both codominance and recessiveness. Cattle. Roan cattle are a product of red and white hairs. Half of their hairs are red and the other half are white. The red and the white genes are codominant to each other. So these kinds of genetic anomalies are pretty common actually in the plant and animal kingdom. Mendel was lucky that he picked pea plants where none of those things were happening. Lastly, before we leave this topic, polygenics is the phenomenon where there's more than two genes that control a trait. Human height, for example, is not controlled by two genes. If it was, we'd either be tall or short. Or if there was incomplete dominance, we'd be directly in between. Turns out there may be 20 genes that control human height. There may be 50 or 100 genes that control human intelligence. There's at least 6 genes that control human skin color. You can see these 3 gentlemen here in this picture have 3 different skin colors. It's hard to get 3 different skin colors when if you just have a Mendel set of genes, 2 genes controlling skin color. That's not going to work. So human intelligence test shows that there are many, many, many genes that control human intelligence, and that's why there's not just smart people and not so smart people or people in between. So polygenics is we won't do Punnett squares with polygenics, but polygenics is very common with a lot of animal traits.